Benbow's on.
Hello, Helen.
Hi, Jimbo.
Hey, Jimbo.
Hi, John.
And good morning or afternoon, everybody who's right on time. We'll give it a few minutes, and then we'll get started about 5 after the hour. Thanks for joining. We'll get started here in about 2 minutes. All right, good morning, everybody. Thanks for joining us today. This morning, we're going to hear from Scilligence's Alan Olague on support for new chemical modalities in Scilligence's Conjugate Builder. My name is Graham Dennis, and I am a consultant here with 2015 Visioneers. Let's see if I can advance the slide. A few quick rules of the game today. We're looking forward to a lively Q&A following the presentation. There are a couple ways to submit your questions, either in the Q&A tool that you'll see somewhere at the bottom of your Zoom window, or you can just pop them in the chat to the host and panelists, and we will pick them up there. I'll sort of be the moderator for the question session, but we'll hear from everybody on our panel that includes other folks from Scilligence. And we do post recordings of these webinars, and they'll be available online for either reviewing or sharing with other folks who couldn't make it. So with that, I noticed we have a nice mix of backgrounds on the call, and we'll spend a minute then now on both the scientific informatics of ADCs and also why they're attractive as drug candidates. So therapeutic modalities beyond small molecule drugs are obviously the norm and have been now for 10 years. But I think the question for folks who come to a call like this is how well has software kept up with the science, right? And candidly, it's not always great. Getting this right in the software has some implications for successful clinical trials. Recently, I've learned about a major pharma's MET inhibitor that reached trials, but an inadequate understanding of mechanism of action led to mistargeting, the wrong bioanalysis, a restart, and a expensive loss of time on its way to the market. And this loss of time in clinical trials, the cost of that has been estimated at 8 figures a day. So not a trivial expense. In our practice, we encounter a lot of clients working with conjugates, polymers, making inventive uses of those tech for consumer products. It's everywhere. ADCs specifically have pharmacological properties such as antibody targeting, their sustained concentration in the plasma and albumin transfer that make them super attractive as drug candidates. Presently, there are 12 approved and 140 in trials, that is ADC drug species. So a few weeks ago, Scilligence did a session with us and shared about their support for HELM. So building on that, they have put a further emphasis on ADC work. So with that, I want to introduce our speaker who will take us through it today. We'll be hearing from Alan Olague, who is the Director of Sales and Marketing at Scilligence, but like many of us, started his career as a research scientist at Bayer BI and Forma, which is about to become part of Novo Nordisk, in both candidate identification and lead optimization roles. And he's been at Scilligence since 2019 in a few roles, and he holds his MBA and studied biochemistry and chemistry. So with that, Alan, I think that's the last slide from me, so I will hand it over to you. And thanks again, everyone, for joining us today.
Thank you, Graham. Right now there we go. Now I can share my screen.
Yeah.
Well, thank you for the kind introduction. And also thank you, everybody, for taking the time out of your busy schedules to attend this talk. So I'm going to go ahead and share my screen and confirm.
Looks good, Alan.
Awesome. Thanks, guys. So as Graham said, late summer, I think in July, Jimbo gave a talk describing our role in becoming the developers of the HELM web editor and the usefulness of the HELM web editor in allowing us to do unique entity registration, not just for small molecules, but also increasingly biologics and more complex macromolecules. So for today's talk, I wanted to give just a brief agenda on what I'm going to be talking about so that you have an idea and hopefully follow along. I'm going to give a very, very brief recap of what Jimbo was talking about, just to set the stage and to explain how we utilize our expertise in the web developer, being a web developer of HELM, to allow to register and get data from more complex drug therapeutics. So in this case, I'll showcase the Conjugate Builder. And in particular, I'll showcase 3 use cases. One with a small drug conjugate, small molecule drug conjugate, an antibody drug conjugate, as you said, Graham. And then we'll showcase how that's utilized in our ELN for registration from the experiment stage to our RegMol module. So for the recap, Jimbo earlier in the summer explained that there's the next generation of therapeutics, right? It's not just small molecules anymore. It's actually expanded dramatically to quite a range of complexity, including biologics. So At the small molecule stage, we still have what I would call a small molecule drug conjugate. For example, Protax, which is being marketed as drugging the undruggable. And I'll go into more detail in a couple of slides on that. But therapeutics now range from peptides, nucleotide-based, antibody-based, as Graham mentioned, antibody-drug conjugates, as well as cell-based therapeutics. And I have to give credit where credit's due. AstraZeneca did a phenomenal job explaining and really summarizing the next generation of therapeutics, which is where I got a lot of my information for this slide. But it just tells you how the complexity of drug discovery is just blossoming. And you need software to be able to keep up with that type of endeavor in the drug discovery field. And so Jimbo described the rise of HELM. So HELM stands for Hierarchical Editing Language of Macromolecules. And it was developed in conjunction with several people. There was a really nice paper by Tianghong Zhang describing the rise of HELM as a notation language for this complex biomolecule structure. Excuse me. And so in this slide, what I'm trying to depict is as you go from increasing complexity from small molecules to macromolecules to biologics, you start to denote that sequence and the capture of informatics changes from the atom level to the monomer level, and then to the increasing complexity of polymer level. And what I'd like to show here is just how it's depicted in our HELM web editor. So we have just the basic atoms that are depicted for small molecules, and you note the increasing complexity as you start to develop monomers, piece them together for more simple polymers, and then really grow to a complex polymer formulation for these types of new therapeutics. And on the right-hand side, I'm just showing the notation that is HELM, that was developed in conjunction with many participants and spearheaded by Tianghong and Pfizer and the Pistoia Alliance. And we were lucky enough to be able to be the web editor for HELM. What I'm showing in this GIF is just a representation of how our HELM editor works in the web. This is our proprietary JSDraw structure editor window. And what it's showing you is just a quick snapshot of how it's building, not just a typical RNA piece, but an RNA strand with unique functionality and unique pieces to it. And as the GIF is moving along, you'll notice that the HELM representation is being delivered in real time. So you can easily string together RNA pieces and modify them and be able to represent them visually in a way that's easy to follow and in a way that's intuitive for the scientist. Which leads us to the next step in the evolution of this. We built the HELM web editor. What's next? Well, there's some criteria that we wanted to develop in order to be able to register this diverse set of modalities and molecular entities. A couple of things that we needed to make sure we had in place before we unveiled our registration system. First, we need to have a uniqueness check criteria for a diverse and versatile set of modalities. This includes chemical structures, biosequences, the proper HELM notation, the composition of bifunctional molecules, as well as unique IDs. If you can't generate unique IDs for your modalities, it's going to be very difficult to track this information and to tie it to the individual drug or development candidate that you're looking for. We also needed to make sure that we had a robust genealogy or relationship network. So we need to be able to track where each piece came from. And you'll see this in a couple of slides as I start to build out the small molecule drug conjugate. You're able to track the origination or the initial inception of that piece as you build it together. And of course, you have to be able to attach metadata to these registrations, right? Images, analytical files, hyperlinks to either your inventory module or your electronic lab notebook, some calculated properties, et cetera. That needs to be captured in this module. And something that is not unique to anybody, we see this quite often, integration capabilities. Most companies have a smorgasbord, right, of different software, be it legacy software that they inherited or homegrown software that they're currently utilizing to try to keep track of all of their entities. So we need to be able to integrate with these unique database connections, either through application programming interfaces or directly to the database. So all of this had to be in place in order for us to be able to to really move forward with what is needed for unique entity registration. And so we have that. It's called RegMol, and it's all under one roof. You can register chemical structures, right, small molecules, and just a variety of modalities that we have here showing in this drop-down window. Polymers, oligonucleotides, antibody drug conjugates. And what I like about this screenshot is that not only are we able to register these things, but we've developed a platform that we can customize to register your unique modality. So this can scale and it can grow to fit the business need. There may be an entity that you don't see here in this drop-down menu. That doesn't mean we can't customize for it. All we need is the feedback, the business need, the logic, and we'll be able to customize that for your workflows. And so we're very proud of our RegMol module. It's a dual system, as I'll explain later what I mean by that. But what I'll focus more is on the registration aspect of RegMol. And it just has this capability to be able to accept different registrations from different modalities, and more importantly, offer uniqueness checks so that you're able to identify, is this a batch 2? Has this been done before? Is this a racemic mixture? Was this antibody drug conjugate using a similar linker? Things that help you move forward in your drug discovery process. So with that, that's a recap of what Jimbo was talking about, and it dovetails into the next step. What I'll focus on today is our Conjugate Builder. And so it leads to the question, well, what exactly are conjugates, right? And they're essentially modalities that take multiple pieces of other entities or modalities and bring them together for a purpose. An example I have here is an antibody drug conjugate, typically used for cancer research, in which an antibody will be used to target a specific cancer cell. Attached to that antibody via a linker is typically a cytotoxic drug or a payload used to destroy that cancer cell. And as you mentioned earlier, Graham, quite a lot of antibody drug conjugates have been put into the market recently. And it's very important to keep track of them and to make sure that the software is there to allow for their discovery. Another one is a small molecule drug conjugate. And the one that I like to use as an example are PROTACs. I think it's, what is it, the I'm not going to remember the proteolysis targeting chimeras. And PROTACs have been heavily marketed as useful tools for drugging the undruggable. And the idea here is that rather than having a cytotoxic drug as a payload to destroy your target, what you instead have is a targeting ligand that'll bind to the specific target, again, attached by a linker system. And rather than having a drug payload, you now have an enzyme, an E3 ubiquitin ligase. That acts as a tag so that your own body then recognizes it and uses its own degradation system, the ubiquitin proteasome system, to essentially destroy that target within the cell and flush it away as waste. And so it's a very unique concept, very successful, and it's quite exciting that we're able to keep track and to help in those new efforts for the discovery of the new PROTACs. The third one that I'll talk about, but I won't have a use case for in this talk, are bispecific or trispecific antibodies. And these are essentially antibodies that are joined together with the hopes that the dual action improves the specificity of the target and again destroys whatever the target is going for. But the reason I highlight these is that we are able to register them and have them in our RegMol application for later registration and perusal. So there are challenges with supporting conjugates at the informatics level, right? And actually even in the drug discovery level. And so the 4 common ones that I'm displaying here are ones that we see over and over again. So the first one is time. Researchers have very busy schedules. The more we can make things efficient for them, the better it will be, right? Time is money. They'd rather be in the lab doing science than in a computer trying to grab different data pieces from different sections and piecing it together. So if we can save time for the researcher by making it either automatic to create conjugates or automatic updating, or simply pulling up complex structures by name or by compound ID, we've done our job. The next thing is the multiple modality handling. It's not uncommon to have a company have several pieces of software for specific modalities. You can have one registration software for small molecules, another one for your antibodies, another one for your cell lines, another one for whatever other type of biologics. And now you have to struggle with piecing together the registration of these different entities into either one place or into an external source where you're trying to make sure and manage that those registrations are up to date, correct, and are not fallible, right, to human error. So that's a major one. How do you handle the multiple modalities? The third is depicting connectivity. You need to accurately predict or depict where your conjugates are actually connecting. Typically, it's a peptide bond, right? It's a different amino acid piece. In the case of an antibody drug conjugate, you have to identify the amino acid that's the connection point in that light chain or heavy chain of the antibody to your linker payload. How do you depict that? That needs to be accurate, and it needs to be easily recognizable by the researcher. And finally, the relationship and genealogy tracking is something we hear quite a lot of when we do client visits or customer requests. How do I track the different components that were used to build a specific entity? And more importantly, how can I see the data with each component highlighted? There may be a perfect linker length for your specific payload. There may be a variety of payloads that simply won't work unless it's tied to a specific targeting ligand. All of that needs to be tracked, and you need to be able to identify the successful conjugates and where they came from and how you can piece them together to do a little bit of analysis and say, well, let's keep this linker length and try to build off of it with our most potent binding targeting ligand, for example. You need to be able to track that in your registration system or your electronic lab notebook. So with those challenges identified, what I'll discuss now are three use cases on how we manage conjugates. I'll start with a simple one, simpler one, the small molecule drug conjugate. I'll move on to the antibody drug conjugate. This is a little bit more complex in that we need more metadata to track for the connectivity. And then I'll showcase via screenshots how these conjugates, can be built with our electronic lab notebook. Okay. All right. So I'll start with SMDCs. So one of the first things you'll note is when you log into our RegMol system, you'll be able to register a variety of molecular entities. What I have here on this screen is a form for small molecule drug conjugates. You'll note in the arrow, our conjugate builder is depicted by a button on the far right section of our proprietary JSDraw entity sketcher. So this is a structure editor that allows you to draw a variety of small molecules, but you can also depict biologics, monomers, and in this particular case, an antibody drug conjugate or a conjugate builder. My apologies. When you click on this button, what you'll see is a dialog box that allows you to compose your small molecule drug conjugate. Now, we are operating here under the assumption that the user has already registered the payload, they've registered the linker, and they've registered their warhead or their targeting ligand. That needs to be put in place before we can actually build the pieces together. If you have not registered these, you will not be able to populate or piece together the small molecule drug conjugate. So this would be a prerequisite. But that being said, we have unique registration IDs for each entity type. You'll notice in our screen, we have a payload compound ID section with the PL003. That would be this compound. We have the linker ID, which is optional, and I'll explain why in the antibody drug conjugate example. And then we have your warhead ID. So all of these can be auto-suggested as you start typing in the letter P, L, you'll see a drop-down menu such as the one that you see here with the warhead ID, and you can select from your previously registered components which ones you want to build. So you can imagine you can create a variety of small molecule drug conjugates just by mixing and matching previously registered components to it. We can restrict these forms by entity type. So a common theme you'll see with our software is that it is configurable and customizable based on the business need and the workflow. So for example, if you would like to restrict this window to only showcase payload IDs, we can certainly do that. On the opposite end of the spectrum, if you want each section to just showcase everything that's been registered, that can also be done if you don't want to be restricted by just an entity type. It really is up to what the needs are for the company. In this case, we have restricted it by entity type, where it's payload, linker, and we're going to call the warhead a small molecule. Okay. Once the components have been selected and we've clicked on OK, what you'll notice is that they'll be displayed on the web editor, and they'll be identified as separate components by brackets. You'll see the brackets separating your warhead. You'll see the compound ID for your warhead, as well as your linker and, excuse me, your payload. What we need to do now is denote the connectivity point. So here it would just be an amide coupling with my primary amine and the carboxylic acid of the linker, and then the primary amine of the linker to the carboxylic acid moiety of your payload. And what you can do is you can actually draw this from our sketcher. So you would choose a single bond and draw it out to depict the connectivity. And the system is intelligent enough to recognize the connection point as you're building out your SMDC. Once you've connected it, right, and I'm showing the length here on purpose just so that you can see where the connectivity point is. You can always clean this up, but the focus on this screenshot is to show you that these are unique connectivity points. If you had different connectivity points on your components, you can choose them as well. And the RegMol system is intelligent enough to track the different connection points. So you will see something like this. This is the creation that you're making based on the components utilizing our conjugate builder. Once the components are connected, it will automatically do what we call relationship tracking. As you scroll down on the registration form, you'll note that the system will be able to tell you which components you use to create this conjugate and the directionality of it. Right now, the directionality is pointing to the left because it's telling you that your small molecule drug conjugate, in this case, I think it's SMDC003, as you'll see on the next screen, contains these three components, and it's built from these three components: your linker, your payload, and your small molecule. But the nice thing is what it automatically will do once you've developed your SMDC, and in this case, it'll show me the relationship tracker that I showed you previously. It will also update the status of these unique entities. So if I were to click on this small molecule, right? So small molecule 001 in this case, it will show me in its relationship tracker that it was used to build small molecule drug conjugate number 3. And the components are updated in real time. So there's no need for the researcher to then go back to the registration system and manually update that they use these components to build this new drug conjugate. It does it for you. Again, time saving, improved efficiency, and it also reduces a lot of error from a manual updating of registrations. So it's quite a unique feature in that this component can be tracked in real time. Okay. All right. Another thing is the relationships can be also detected in our assay views. For those of you who are not familiar with our RegMol module, it's really a dual functioning module. It's not just used to register unique entities. It can also be used to create protocols, register those protocols within the system, and then upload assay data from those protocols based on unique entities. When you do so in RegMol, right, it can handle the assay data on those unique entities. You can create what we call assay views. The assay view is your own unique data analytic viewpoint of a particular protocol with a particular set of compounds. And what I'm showing in this screenshot is a built-out assay view that contains this small molecule drug conjugate, and it shows the entire structure as well as the different components that are related to it and other metadata. So we can see the molecular weight, the potency in IC50, the volume of distribution, the clearance, and the half-life of the compound. So you can imagine having a list of these small molecule drug conjugates and being able to determine just by potency, for example, if there is any activity that's linking it to either the ligand, the payload, or the linker length. And it's a quick snapshot to give you a little bit more insight into your SAR analysis. So again, the power of RegMol is not only does it capture the uniqueness of that small molecule and the components of that drug conjugate, it can also associate specific data related to this drug conjugate. Okay. So that's small molecule drug conjugates. We'll get into a little bit more complexity with antibody drug conjugates. Now, when you click on the antibody drug conjugate builder, same button location on the structure editor, but you'll notice that in this case, the dialog box has a couple of extra fields associated to it. Excuse me. You'll still contain your 3 sections. In this case, it'll be an antibody identification, linker ID, and payload ID. The reason you see optional under linker ID is you can actually create a payload ID that already contains the linker. So it'll be your payload linker. And that way you will not need to put in a payload ID. It would simply be the antibody connected to the payload linker ID. For the purposes of this talk, I'm showing the linker as an actual field. You'll note that the name is PEP. We use that for peptides. So this is going to be a peptide linker that is used for this antibody drug conjugate. And the antibody has also been previously registered as well as ANT007. I'll show you on the next screen that contains the actual sequence of the antibody. So it's not just a number, it actually contains all the information for the antibody, as well as the ability to annotate where the linkages are located. But we also have here, the antibody position where it's located, right? So in this case, it's on the lysine, or the connectivity point will be on the lysine. And then we have the DAR, the drug antibody ratio. In this case, we have a drug antibody ratio of 3. So 3 entities to 1 antibody. It'll be a 3-to-1 ratio. Once we've identified these 3 pieces, we can build the conjugate and move on to the next screen. You'll note, similar to the small molecule drug conjugate screenshot that I showed earlier, you'll have segregated the 3 distinct pieces of your antibody drug conjugate. The antibody, which it's not just a graphical display, but it actually contains the sequence that you can put in there from when you registered it earlier. And what we have here are the highlighted lysine points where the connectivity is. And we see here the position is the kinase connectivity with a DAR ratio of 3. This is a typo. I meant to highlight the K. And then you also will now have your linker designated in this screenshot, as well as your payload. The unique thing about this is that the information is populated with different languages and different syntax. The antibody has the sequence information. The linker is displayed as a peptide using our monomer display, right? This utilizes HELM. And then of course your payload is displayed as a structure, a small molecule structure. So our JSTRAW structure editor is able to handle all of this in real time with one display. Shows you the power of HELM, and it shows you the capability of our structure editor to be able to accommodate and truly be a unified platform for not just small molecules, but for biologics. What I have here, and my apologies for the garish colors for the highlights, I wasn't able to change those in time. But what I have here is how do we then identify this antibody drug conjugate, right? There's a lot of information packed into this compound ID that we utilize in order for the researcher to be able to, at a glance, identify what was being done, what components were being used when this drug conjugate was assembled. So in yellow, you will have your prefix, which is just ADC for antibody drug conjugates. In this light purple, I'm not good with colors, magenta, I guess. In magenta, you'll see the payload is the first thing that's identified in the conjugate ID, and that would be this section over here. This is then followed by the linker. So we can identify that this was a linker, in this case, linker 10. The payload was SCI 110. The linker is peptide 10. The antibody that was being used was antibody 7. If you remember when we registered it earlier, it's the 7th antibody that you had registered. And then your lysine connecting point is the next thing that's identified, excuse me, in this type of ID. The DAR ratio of 3 is also indicated at the end of the compound ID. And the -1 indicates that this is your first batch of creating this antibody drug conjugate. So if you were to create the exact same ADC and register it again, it will be able to recognize that this has been created before and will then utilize a batch 2, batch 3, batch 4, etc. You'll be able to then identify the uniqueness of this ADC. Okay. So I'm going to switch gears now. And sorry, I didn't have a transition slide. We're going to then use this into ELN. How can we utilize our electronic lab notebook to be able to create these conjugates? You'll notice that the syntax here is a little bit different, and I did that on purpose to show you that we're able to handle different types of syntaxes and how they can be registered into Regmol. And what we have here on this screenshot is an ELN, an experiment, an electronic lab notebook experiment that already has an antibody designation and a payload linker entity, right? So we have already identified the bifunctional nature of this ADC, and we've connected this payload linker. This payload linker has been registered before. So if you remember earlier on when I said that linker was optional, if you do something like this in the experiment where you've already registered the payload with the linker, then there's no need to identify a linker in the ADC. So what we're going to do is we're going to connect the antibody to the payload linker, and it has that data. You'll note you do not have to draw out this entire macromolecule. If you simply type in the sample ID of the macromolecule, you'll pull in that information from Regmol into your ELN. So this saves the researcher time, because I could imagine it would be a nightmare trying to draw this out by hand or using our sketcher. No need to do that. You register it once, you pull it in, and you're good to go. The antibody is a unique antibody that was populated in here with the light chain and heavy chain sequences, and we were able to identify the component of each of these reactants, right? In this case, we have a column that says, what type of component is this? We've identified this as an antibody and the payload linker. Now, the next slide shows the product ID. Once I've created the product, I have a unique registration number, and you're also able to manipulate the molecular weight. So you cannot determine the molecular weight automatically with our software. That's typically determined in an experimental format using high resolution, I guess, mass spec, that you would be able to determine this molecular weight. But this is editable. Once you put in the molecular weight, it'll give you an estimate or a summary of the DAR ratio. A DAR ratio of 2.5 here indicates not that you have exactly uniform 2.5, payload linker to antibody ratio, but it's a combination. You can have a 4-to-1 ratio, 3-to-1, 2-to-1, or even just one payload linker to antibody ratio, and it harmonizes it into a summary of a 2.5 DAR ratio. You'll note too that the registration number is a little bit different from what I just previously described. This has to do with the fact that we had already registered this payload linker as a scaffold. So this is scaffold 3, an antibody 18, and there's a different rule set for this type of nomenclature. I can't remember it off the top of my head right now, my apologies, but we're able to customize different rule sets for your compound ID registration. So if you don't like our out-of-the-box structure compound ID for antibody drug conjugates, we can work with you to customize it so that it makes more sense to your business needs. And of course, once you've identified the molecular weight and the purity and the ratio, it can give you the theoretical yield based on the amount that you've isolated from your ADC. Excuse me. And so I'm going over time. To wrap it up, we set out to the evolution from Jimbo's earlier talk about HELM and how we are the developers of the HELM web editor. And then we set out to do a unified entity registration module and software capability. And I'm hoping that I was able to show you that. The key impression that I want to leave and to the audience in this talk is that our platform can be customized to fit your need. So if you do not see the entities on the drop-down menu that you're looking for example, I think we do have polymers here, but let's say you're working with polymers. Actually, there are polymers here. We can customize the platform to fit in your business need, because all it really is just piecing it together and applying the uniqueness check rules that we already have built into the system to fit your commercial need. So we're very proud of Regmol. It's a very powerful tool. It has a lot of complexity built into it, but it also has a lot of flexibility capable for our team to use to fit your workflows or your business needs. And with that, I wanted to thank you for patiently listening to the presentation. Thank you very much.
Hey, thanks, Alan. That was terrific. And I just want to comment, Scilligence has come a long way. This is really cool to see. And I feel like y'all could do a whole nother session on, I noticed that cell line registration is present in Regmol, and there's probably a whole other session out there on that would interest a pretty big audience. So thank you very much. It's tough, can be tough to talk into your computer continuously for half an hour. So we really appreciate that. So questions are flowing in, and I'll just get going. First one, and candidly, this is from me and John. Are there concepts of, well, there's certainly going to be concepts of substructure searching in these more complex polymers or combinations, right? I was wondering if you could comment on that. And then also is, in small molecules, we talk about a similarity search or like a Tanimoto score type search. Is there an analog to that in this broader world of registered entities?
No, so I'll attempt to answer this, and then if my fellow panelists want to chime in and elaborate on it, I'll let them go. But yes, you bring up a great question in terms of substructure search. So we have a robust searching, advanced searching mechanism throughout all of our modules, right? So Regmol is no exception. If you want to do a substructure or a similarity search, you simply click on, there's an icon with a little magnifying glass that's advanced search. You'll put in the substructure, and it'll scan the entire database to populate the results of that substructure search. The same can be said for similarity search. If you want to get a little bit more complex, right, where you want to do Tanimoto searches or what's the other one, Merco scores, et cetera, for perhaps high throughput screening, cluster identification, that gets a little bit more complex. And in that case, we would have to work with you to try to identify how can we apply those types of searches either within Regmol, or how do we export the information that we have in Regmol to either another software that you're currently using for data analysis and visualization for that. So innately, we don't have out of the box built in the Tanimoto and the specific search requirements. But again, that's the beauty of Regmol. We can work with you to either customize to make it work, through Regmol, or if we realize that, nope, this requires a complete overhaul of our coding, then we look for the alternative, right? If you have another system that's generating these scores for you, how do we integrate that with it, right? So you can have third-party systems. We partner with D360, for example, for data analysis. If you're using, I don't know, Schrodinger's live design to do molecular modeling, and it already populates these type of scores for you, how do we export that data from Regmol as a source of truth into your system? So it can be done. And I see Jimbo's chiming in. So go ahead, Jimbo.
Yeah,
Yeah,
please.
I think that's an excellent questions. I think it's the purpose of a Scilligence solution is really I would say following the fair data principle. And so your question is related to substructure search. The feedback I want to add to this questions is basically what can you find, when user actually do a substructure search in the context of conjugate builders. So basically, when user do a search like that, they will not only find the payload, the linker, but also all the conjugate containing that substructures. If your linker is actually represent in, I would say, a form of macromolecule in the HELM structures, because HELM structure containing, unnatural amino acids and unnatural nucleotide, that substructure search only find, those modality as well. So I think the key thing is really the core technology behind the indexing. So that enable substructure search to find all the modality that containing that substructure. Just want to kind of add to what Alan has already provided in terms of feedback. Yeah.
Awesome, thank you, Jimbo. A couple really interesting ones popping up here, and I just want to say, Bill, I see your questions, and we'll definitely get to those. Those are great. But first, this is interesting. You mentioned PROTACs, and a PROTACs could just be registered as a small molecule, like they have been for almost over 20 years. Is there an advantage to using Conjugate Builder that you showed to registering a PROTAC over just registering it as yet another small molecule?
That's actually a great question that we've been asked before, and I don't work in the PROTACs industry, but my answer would be yes, in terms that, in the essence that you can identify the advantageous or the more potent section that's leading to your PROTACs compounds. So what do I mean by that? PROTACs involves different E3 ligases, right? VHL, I forget the other one, CRB, different linker lengths, and then of course your different small molecules that are attaching to it. So as you're running and you're testing and you're doing analysis on these PROTACs, you start to notice hits and potency. Now you can, if you register using the Conjugate Builder, you can create an assay view, as I showed earlier, of your most potent hits, and you can start to denote a pattern. Is there a certain linker length that is optimal for this particular target? Is there a certain E3 ligase that we're using that is more successful in destroying and producing the effect that we want? Is there a small molecule, do a little bit of SAR analysis that we can then identify and use to create more elaborate combinations of PROTACs? You can do that using the Conjugate Builder. If you don't use the Conjugate Builder and you register them in its entirety as the bifunctional molecule with the ligase, the linker, and the small molecule warhead, you can still see the potency and try to analyze or try to identify where the commonalities are, but it won't be as straightforward as what I showed earlier with that assay view, in which you can separate the components of each, for example, PROTAC, and then do that further analysis. So I would think it would only help with the analysis, and you can identify the optimal piece that you're trying to look for that particular target. Of course, we all know that changes with targets. One kinase inhibitor is very different from another in terms of specificity and what you're trying to attack, but you can certainly use that tool set to enhance the analysis set data by using Conjugate Builder.
Yeah, I think I would like to chime in on this question too. I think it's very interesting questions in the way that, right, I think maybe some of the audience here are medicinal chemists. Okay? I think, we analyze SAR based on memory in a traditional way. Okay, that's why, I think as Graham mentioned that, PROTAC has been registered as small molecule, in the form of small molecule for, 15, 20 years, right? And but when you do the analysis of structure activity relationship, is you have to really base on your memory of all the potent molecule has ever been generated for that program. But with the conjugate builder, it's automatically delineate the lineage of components, relationship between a component and it's the parent PROTAC molecule. So that is really help you to do structure activity relationship analysis. And with the, I would say the advance of, I would say, machine learning and artificial intelligence analysis, now you have a basic set of rules you can set, actually, maybe using those informatic tool to help you, to do those analysis as well. So this become a machine learnable type of arrangement in a database setting. Yeah. Okay. Yeah.
Cool. Thank you, Jenba. So stick around. In your, I sort of give this question, it's the best question award. In your SMDC example, there were more than 3 lysines present in the sequence. Does the user specify which lysines are available for conjugation? And related, does Regmol identify surface available amino acids?
I got to
Maybe
unmute myself.
I mean, for these questions, yeah.
Yeah,
Yeah.
go for it, Jim. I was going to say, I think it's in the
Maybe I can time
ADC, not the SMDC, but that's okay. Yeah.
Yeah. So so let me help answer these questions. There's 2 modes for building a conjugate that our team has built over the years based on our pharma biotech customers' requests. One mode is that user would define the connection point. So in this question, Bill asked, well, there are 3 lysine in the linker. The example that Alan showed is that the user, when they connect the linker to the payload, they actually define, where's the connection point. So as a chemist, actually, you define what bond will be broken and what bond will be formed. Okay, so that's the manual mode, and that handle conjugation building one at a time. I think Bill's question is also related to a second mode that is a little bit more sophisticated now, is when you handle a bulk registration of a conjugate, you really don't want to build those connections, one at a time. So how do you define business rule that automatically identify what, I would say, counterpart for the conjugation is, and then how to do the bond breaking and forming and using business rule. So that is actually handled in a bulk mode, bulk registration mode. And that is a little bit more sophisticated. So that's why it's not trivial actually to kind of build a conjugate builder that can handle all the scenario. Yeah. To follow up on the question, can you identify surface available amino acids using Regmol? At the moment, Regmol doesn't do that function at all. And it's really, especially for antibody, residue, right? What is actually conjugate to the linker payload? It's actually user defined. So, if you have a cysteine, which is, you can have 2 different modes. One is non-specific conjugation. One is site-specific conjugations. So those will have to define by the scientists. Yeah, Regmol doesn't do the automatic, searching and identifying. Yeah.
Yeah. Cool. So 5-minute warning. But let's see how many more we can answer. This is a neat question. You gave some examples, and of course in these notations for biomolecules, we use some contractions, right? Or some coding. Do you define that chemistry, or I'll ask it exactly as the asker asked it. Where is the chemistry defined? The chemistry that defines how the antibody links to the linker and how the linker links to the small molecule.
So that can be defined in Regmol. If I'm understanding the question correctly, I'll give an example. We have a monomer library, right? And with that monomer library, we can designate different connection points, R1, R2, et cetera, for, say, a specific peptide. You can define, as you're registering the compound, you can define where that is going to be connected, if there are multiple connection points, a pop-up window will appear and say, where do you want to connect this amine to this carboxylic acid, for example. That monomer can be defined in Regmol. It can be user defined. We typically reserve that for an admin purpose so that there's not a smorgasbord of different monomers, but you can certainly edit the connection. Let me see if I can show you in the GIF. Actually, no, I do have a backup slide for this. So we have monomers here that we can display. These are controlled in Regmol. You can create these unique monomers in Regmol. In this case, we're highlighting peptide monomers. And if you go to the structure view, you'll notice the uniqueness point. So for example, in the structure view, we know there's an R2 designation here. If there were R1 and R2, you can actually choose as you draw that bond to connect from one area to the other. A pop-up window will appear that will ask you, where do you want this connection point to be created? And that is controlled in Regmol. I'm hoping that answers the question. Jimbo, you can chime in.
Yeah, I think what Graham asked, if I understand Graham's question correctly, is really what is the business rule to determine, a molecule is unique in the database, and I think conjugation, location, what conjugations, I would say, chemical group actually form. So those are the things that that is actually fairly unique in Scilligence technology. We actually build a hash code, a algorithm that can take into all the factors that need to be taken into consideration to determine the uniqueness of molecule. So we have customer run into problem of just simply using HELM string as a uniqueness check criteria. And that actually create molecule that, they're the same molecule, but their HELM string is slightly different. And that actually have multiple ID corresponding to registration ID corresponding to the same, the same molecule. So we certainly don't want that to happen, because I think, registration system has to be a one-to-one relationship between the identity of that molecule and the registration ID. So that is actually taking into a lot of consideration to build that proprietary hash code generations technology. Yeah.
Okay. 2 minutes. So no pressure, but maybe sort of lightning round. I think this question is about the extensibility of the HELM definition. It says, you mentioned assay stroke metadata are directly associated with the entity in Regmol. Is that data structure integrated directly into HELM? And again, that sounds to me like extensibility of HELM. Or does HELM include those assessments as part of the specification?
Yeah, maybe I can help chime in. I take the lead to provide feedback
Sure.
to these questions. So this is not related to HELM, what Alan is showing the assay view. So Regmol is actually a two-in-one application.
Yeah.
One side of the Regmol is handle molecular registration or entity registrations. And the other side is actually handle assay protocol management. So basically, analysis management. we use it in as assay protocol management, but you can actually manage all the analytical, even the the GLP analytical protocol as well. So basically the other side is help. It's a assay registration system. Allow company to register all the analysis methodology, and also capture all the analysis data. Can be assay, can be chemical and analysis, can be biological analysis into the database, so that all those metadata from assay or analysis will be associated with the protocol. If there are certain molecule, including HELM-based molecule, that have been tested using those protocol, those data will be linked to those molecule via the registration ID in the database. So that is a relation database to connect, I would say, the source of truth together. How you're going to look at the data? It's really, we provide a lot of options So you don't have to use Scilligence tool. You can use any data analytic tool to pull those data, registration data and assay data into into any tool of your preference for data analysis and visualizations. Yeah.
And, of course, frequently that data we're going to want linked at the batch level, right? So it really has to reach deeper into the data structure. And honestly, I think there's another hour to talk about batch handling against this plethora of entity types, right?
Yes.
We're already a tad over, and I know we want to stick to time, but I want to get to Prashant's question, where he says, are there best practices in data strategy and management for the complexity and volume of data generated separate from system strategy to support research or operation KPIs? And I'll start this one just from a 2015 Visioneers perspective. We are super enthusiastic about the FAIR data principles, that data should be findable, accessible, integratable, and reusable. So Prashant, I would sort of point you in the direction of the FAIR project that is led by the Pistoia Alliance. So find that on your favorite search engine, or come to 2015 Visioneers website, or I'm sure Scilligence has thoughts on the importance of FAIR, because that's in your question. I really see, how do we make the data meaningfully useful in our programs, right? And that's about FAIR. It's sort of a nice note to conclude on. I'd love to hear from Jimbo or Alan if y'all want to add on to that.
Yeah, I can chime in. I think Prashant has excellent questions. I think one suggestion I have is about best practice in data strategy is actually adoption of standards. So, I think, coming back to HELM, this is developed as a standard to bridge the gap between structure-based modality versus letter-based sequence modality in biologics. So, we have actually worked with customers using their own homegrown, standard, and it's make it very difficult. Every company is different, so they cannot talk to each other. They cannot collaborate because they don't understand each other's language. So I think adoption of standard early on, I think it will be a good considerations of best data practice. Yeah.
Awesome, thanks, Jimbo. And it is an active versus passive effort. There's some laws of the universe that describe, our tendency toward chaos. And probably an honorable mention for data governance there too, but that's another webinar. So we'll wrap it up here. Thank you so much, Alan, Jimbo, Aaron Moran, John Conway, and our whole team for pulling together for this session. We'll make this available on the 2015 Visioneers website, 20visioneers15.com. I'm certain Alan will get it onto Scilligence.com as well. And
of course.
Yeah, and so we'll look forward to seeing you next time. And thank you so much, everybody, for joining us. Have a great afternoon.
Thank you, everyone. Take
Bye.
care. Bye.